Welcome to online classes DO Nalgonda. Children, we observe waste everywhere in the schools, in office, in our house, even in factories. Wastes are generated. So these wastes have to be disposed of, or else it can cause harm. So in the same way, even in our body, wastes are continuously getting generated. Those wastes have to be removed from our body, otherwise it results in a diseased condition, or it can also be fatal. So what are the different kinds of wastes which are generated in our body, and which organ system is involved in cleaning the waste? So our lesson is going to be excretory system, or the waste disposal system. Excretory So there are various kinds of wastes which get generated in our body, that is the gaseous waste, solid waste, and nitrogenous waste. The gaseous waste are removed while exhalation. The carbon dioxide is getting removed while exhalation, and the solid waste are adjusted out, that is the undigested waste are adjusted out through the anus. And the nitrogenous waste, this is what requires the excretory system. So what is this nitrogenous waste, how it is generated, we are going to learn in this chapter. So the nitrogenous waste, these are in the form of ammonia, urea, uric acid, creatinine, etc. So these are the different kinds of nitrogenous waste. Where are they generated? They are generated in each and every cell. It is generated in each and every cell. How is it being removed? It is the blood circulatory or the circulatory system collects all these waste from each and every cell and passes it on to the excretory system. So how these nitrogenous wastes are getting generated? Let us see. You know, living organisms requires constant energy, for which they breathe in oxygen and they eat food. The food gets digested, it is being absorbed, and energy is produced. So that is produced because of reactions, thousands of reactions which occur in our body, and those reactions are like anabolic reactions, catabolic reactions. Anabolic reactions are the building up reactions. Building up reactions means smaller molecules are brought together to form into a larger molecule. Smaller molecules together form into a larger molecule. Example is photosynthesis, which you have learnt in your previous lesson. And catabolic reaction is breaking down reaction, wherein larger molecules are broken down into smaller molecules. Larger molecules into smaller molecules. Example, respiration. Like these, all the reactions which occur in our body are either anabolic reaction or catabolic reaction. There are thousands of reactions which keep on occurring for protein synthesis and lipid synthesis, and there are several reactions. All the reactions put together, a sum total of all the reactions is called as metabolic reaction, is called as metabolic reaction. So these metabolic reaction produces various kinds of products. You have learnt in chemistry, reactants form products. So various kinds of products are being formed. The products are either useful or harmful. The ones which are useful is used in the energy production and for different activities, and the products which are harmful and the products which are in excess has to be removed from our body. And there comes the activity of the excretory system. In the human body, the excretory system is made up of kidneys. Kidneys are the excretory organ. In the human body, the excretory organs are kidneys. So kidneys help in removing the waste from the blood. So there should be a proof for anything. So in the textbook, in page number 76, you can see blood and urine analysis, wherein you can find different substances which are present in urine as well as blood. Can you just open up page number 76? There you can see in the blood reports, you can find glucose, sodium, potassium, chlorides, urea, creatinine, uric acid, cholesterol, triglycerides, calcium, phosphorus, proteins, albumin, etc. And in the urine report, you can find same substances of blood is also found in the urine, except few like glucose, cholesterol, triglycerides, bilirubin and albumin. These are not found in the urine. In case if they are found, that means it is because of some disease or the other. It is an indication of some health issue or the other. Okay, now let us get into the details of excretory system. Welcome to online classes DO Nalgonda. This session is in continuation with the previous session on the excretory system, especially the human excretory system. The human excretory system mainly comprises of a pair of kidneys, a pair of ureters and urinary bladder. Firstly, we will learn about the kidneys. Kidneys are bean-shaped organs which are reddish brown in color, and they are located in the abdominal cavity on either sides of the vertebral column, and the outer part of the kidney is convex and the inner part of the kidney is concave in shape. On the above the kidney, you can see this orange colored structure which are nothing but the adrenal glands. Now let us learn about the dimensions of the kidney. The kidneys, as we have already learned, that it is bean shaped, and the length of the kidney is 10 centimeters and the width of the kidney is around 5 to 6 centimeters, and the thickness of the kidney is around 4 centimeters. So you can see there is a notch in the kidney which is called as hilus, from where the renal artery, the renal vein and the ureters arise. Now we'll just look into the structure. Here you might have observed that the right kidney is slightly lower than the left kidney. This is called as the right because we are seeing the opposite side. So this is right and this is the left kidney. Right kidney is placed slightly lower than the left kidney. It is because of the presence of the liver above. As the liver is present here, the kidney is the right kidney is slightly present lower than the left kidney. Okay. Now let us look into the internal structure of the kidney. You might have observed the difference here. This looks plain and this looks somewhat with some details, right? That is because of the section. When we take a longitudinal section of kidney, it looks like this. Let us look into the internal structure of kidney in detail. You can just observe two different regions in the kidney here in the internal structure. You can see two different zones. The outer zone is dark in color and the inner zone is light in color. The outer zone is called as cortex. It is called as cortex, and the inner zone is called as medulla. It is made up of medullary pyramids, and the medullary pyramids, they lead to a conical structure called as calyces. These brown colored structure which you can see are calyces. These calyces leads, each calyce from each pyramid leads to the pelvis. This region is called the funnel shaped structure is called as pelvis. Okay, and you can just see in the pyramids white or silver colored streak like structures. Those are nothing but the nephrons or uriniferous tubules, which are the basic unit of kidneys. These are the filtering units of kidneys which helps in the filtration of urine from the blood. As you can see, there are two tubes which are entering inside the kidneys, which is red and bluish in color. The red one is called as the renal artery. The renal artery is actually carrying, it is a part of the aorta which is carrying oxygenated blood, but it is fully loaded with waste. It is carrying the blood which is loaded with waste. It enters inside the kidney near each uriniferous tubule or the nephron. It divides and redivides, forms capillaries and gives out the waste into the uriniferous tubules, and they join back to form venules, and venules join back to form renal vein. So the renal vein is carrying blood away from the kidneys, and the blood which is carried out is deoxygenated, but it is free from the waste. Children, as you have seen, these white colored streaks which are called as uriniferous tubules or nephron, let us learn about the structure of nephron in detail. This is how the structure of nephron looks, and it looks complex, but don't worry, I'll make it easy for you all. You can observe, you just need to observe these orange colored structures initially. only this orange colored structure. The cup shaped structure, which is a blind cup shaped structure, is called as Bowman's capsule and glomerulus. This whole thing is called as Malfigian body. So nephron is basically divided into Malfigian body and renal tubule. So this part is called as Malfigian body, and the remaining part of the orange colored tubule, which you can see, is called as the renal tubule. So let us first learn about the Malfigian body. The Malfigian body is made up of a cup shaped structure which is called as Bowman's capsule. It is a blind cup shaped structure, inside which you can find a bunch of blood vessels or blood capillaries, which is called as glomerulus. So how is this bunch of glomerulus is formed? Let us see. As you have seen in the previous figure, the renal artery which enters inside the kidney, the part of the renal artery which is here divides and forms into an efferent arteriole which is carrying oxygenated but blood which is loaded with waste into the Bowman's capsule. Here it divides and redivides to form a network of blood vessels. Again they combine to form efferent arteriole and comes out of the cup shaped structure which is called as the efferent arteriole. You can see the structural difference here, the difference in the diameter of efferent arteriole and efferent arteriole. Efferent arteriole is slightly thicker and efferent arteriole is slightly thinner. So it is just not present like that. There is a purpose for that. The thicker blood vessel here allows more blood to pass, and as the blood vessel narrows, it creates a pressure through which the blood filter out the filtrate, and as the passage is very narrow here, pressure filtration occurs. So from here the efferent arteriole, it moves up and it spreads out to form a network of blood vessels which are called as capillaries, and these capillaries, they join together to form venules, and it comes out in the form of renal vein. This renal vein, again you can see it here, they join together and come out of the kidney. Okay, now let us see the next part of the nephron, which is renal tubule. So renal tubule begins from here. This whole thing is renal tubule. So let us see the parts of the renal tubule. Renal tubule is divided into three parts. The first one is PCT. The second one is called as the Henle's loop, and the third one is DCT. PCT is nothing but proximal convoluted tubule. This portion is called as proximal convoluted tubule, and next to that the U-shaped structure which is called as Henle's loop, and the next portion is called as the distal convoluted tubule. The proximal convoluted tubule is called so because the name itself indicates pro means first. So it is placed first. So proximal, it is convoluted, right? That's the reason it is called as convoluted, and it is in the form of a tube. So it is tubule, and this next to that is Henle's loop, which is based on the name of the discoverer, scientist Henle's. And the next portion is called as the distal convoluted tubule. Distal because it is placed at a distance. So it is called as distal convoluted tubule, which opens into a duct called as collecting duct. This duct is common for 2 or 3 nephrons. So through 2 or 3 nephrons, distal convoluted tubule can open into this collecting duct. Cut. Now let us look into the process of urine formation. Urine formation occurs in 4 steps. The first step is glomerular filtration. Or this is also called as ultrafiltration. The next part is tubular reabsorption. And the third one is tubular secretion. And the last step is concentration of urine. So the first step, which is glomerular filtration or ultrafiltration, occurs in the Bowman's capsule. There you can see the efferent arteriole, which divides and redivides to form a network of blood vessels, which is called as glomerulus. As I have discussed earlier, the thickness of efferent arteriole, the diameter of efferent arteriole is more than the efferent arteriole. This creates difference in pressure. As the blood vessel becomes narrower, the pressure inside this increases. As a result, the fluid whichever is present in the blood, except the blood vessels, almost all the fluids drain out of the blood vessels and they reach the cup-shaped Bowman's capsule. So here the inner lining of the Bowman's capsule is made up of a special kind of squamous epithelium, which is called as podocytes cells. They are made up of special kind of epithelium, which is a squamous epithelium, which is made up of podocytes. These podocyte cells are porous, which has lots of gap in between, and it allows the draining, or it forms the conduit for the filtrate to be collected inside the into the walls of the Bowman's capsule. That's how the primary urine or the primary filtrate is reaching the Malfigian tubule. Now, after it reaches here, the primary urine is passed into the proximal convoluted tubule. The urine whichever is formed here, the primary urine or the filtrate whichever is formed here is very dilute in nature, as most of the liquid or the fluid from the glomerulus is filtered out. So it comprises of lots of water and lots of useful substances along with the waste. So the urine whichever is formed here, the primary urine whichever is formed is very dilute. So the dilute urine reaches the proximal convoluted tubule. The proximal convoluted tubule is again covered by this peritubular network of blood vessels which is formed from the efferent arteriole. So as we have discussed that the primary urine which is formed is very dilute with essential substances, what happens is the next step, that is tubular reabsorption occurs in the PCT region, where all the necessary substances are reabsorbed. Now what are the necessary substances? They are glucose, certain amino acids, vitamin C, some salt ions like potassium, chlorine, calcium, etc. are being reabsorbed. That means again it is going back to the blood flow from the PCT, from the renal tubule. It is going back to the blood vessels. That is the reason it is called as reabsorption. From the tubule, it is reaching the blood flow into the peritubular network. Now, most of the water, 75% of the water is being reabsorbed in this region. Which region? It is in PCT, and the urine is getting concentrated. Now, the concentrated urine passes into the Henle's loop. This is the descending portion of the Henle's loop, and this is the ascending portion of the Henle's loop. Now the filtrate passes down the Henle's loop. Meanwhile, in the Henle's loop, 10% more of water is being absorbed, making the urine more concentrated. Now, in the region of DCT, distal convoluted tubule, the filtrate is reaching, and here again you can see the peritubular network is associated, right? So the urine is concentrated, but still some of the waste are lying with the blood. So that should be secreted back. That is nothing but the third step, tubular secretion. So tubular secretion occurs in DCT, in distal convoluted tubule, where all the unwanted substances or the waste substances which are present, which are reabsorbed, have to be pushed back into the tubule again. That is called as tubular secretion. Let us see what are the things which are being sent back from the blood vessel into the tubule. Okay, they are urea, uric acid, creatinine, certain salt ions like potassium, sodium, calcium, and chlorine ions. So these ions and urea, uric acid, creatinine, all these are the waste which are passed out of the blood vessels and it reaches the distal convoluted tubule. Now, in this way, the urine is brought to proper concentration and proper pH level, which is passed on to the collecting tubule, In the collecting tubule, further concentration of urine occurs. So, the concentration of urine which occurs in the collecting duct. Here there is a hormone which is called as vasopressin. The blood vessels releases this hormone which is called as vasopressin. This hormone is produced only when we drink less water, when the body has to produce concentrated urine, especially in summers. In case if we don't get water, we pass out concentrated urine. So, as we can't afford to lose more and more water, here vasopressin comes into action to release only less amount of urine. In patients, those who are suffering from diabetes, this hormone does not work, or this does not get produced. In especially diabetes insipidus patients, this hormone does not get produced and they pass frequent urine and large amounts of urine. Now, let us see how this nephron is arranged in the kidney. You can just observe the upper portion, that is the Bowman's capsule, the PCT and the DCT, which are present in the upper portion of the nephron. They are arranged in the corticular region, or in the cortex they are being arranged. The Henle's loop portion and the collecting duct, they are placed in the pyramid, medullary pyramid. So, you can just observe these lines. These lines are nothing but mostly made up of collecting ducts. And even the Henle's loop, the U-shaped tubular portion of the nephron, is also deeply seated in the medullary pyramid. And even in this diagram, you can see the same streaks, which are nothing but the collecting duct, which helps in the formation of micro droplets of urine. These droplets, micro droplets of urine, they get collected in the calyces and it is sent to the pelvis region. All the micro droplets of urine, it gets collected and forms a large droplet of urine at the junction of the pelvis and the ureter. You can form a large droplet of, you can just see the large droplet of urine here. Okay, thank you.